Thank you all for joining us this morning. Thank you for coming to this seminar. So basically what we're going to do this morning is we are going to summarize two recent studies that we've just conducted. Then we're going to have a series of questions for our expert panelists. And if time permits, we will go ahead and open up the questions to the audience. So first, I'd like to introduce our expert panelists. On my right is Dr. Tom Chambers. Dr. Chambers is a professor or a professor at Gluck Equine Research Center at the University of Kentucky. He's a leading researcher in respiratory viruses, specifically equine influenza and herpes. He's a member of the OFFLU-OIE, which is basically the World Health Organization for animals that tracks and monitors influenza outbreaks. Next to Dr. Chambers is Dr. Tom Divers. Dr. Divers has a lengthy resume, but Dr. Divers is a professor of internal medicine at Cornell University. And Dr. Divers is widely published, has numerous awards, so he doesn't need much more introduction than that. Next to Dr. Divers is Dr. McKenzie. Dr. McKenzie is an associate professor of equine medicine at the Virginia, Maryland College of Veterinary Medicine. Previously at the Marion DuPont Scott Equine Medical Center, so he's been basically a Virginia boy for most of his academic career. I'm Dr. Mark Crisman, previously professor of internal medicine at the Virginia, Maryland College of Veterinary Medicine, and currently a senior veterinarian on the equine technical services team with Zoetis. We have numerous choices available to us when it comes to vaccinating our horses. Many of these newer polyvalent vaccines that have come out on the market really within the last 8 to 10 years have provided tremendous convenience to our vaccine protocols. However, one of these concepts is this idea of antigen interference. And this is a real concern that really has been critically evaluated on the human side of the equation, specifically pediatric vaccinology. And it's also been evaluated in cattle and some other species. But this concept has been largely overlooked on the equine medicine side of the equation. So essentially, what this is, this idea of antigen interference, what the FDA has stated is that a multivalent vaccine should ideally generate a protective immune response to each of their individual constituents that's equivalent to that achieved when they are administered as a monovalent. So to that end, we have looked at several studies, but the bottom line is that the optimal immune response depends both on what is in the vaccine and how that vaccine is specifically delivered. So we've done several studies along that. We've done these studies over several years. Some are published and some are in preparation right now. But we've done a couple of studies looking at influenza, and essentially these studies were designed to determine the serological response of equine influenza when it's administered either as a bivalent, so influenza with herpes antigen, or co-administered with a separate core antigens, and then also administered as what we call these big combinations, or the flu herpes with all the core vaccines in a single syringe. So these studies usually involve 20 to 40 seronegative horses in the group. Basically, we had serum drawn at the days you see on the slide, and they were evaluated for their titers against various clade 1 and clade 2 strains, circulating strains of influenza. We looked at all three major manufacturers, so this was Zoetis, Merck, Boehringer, And the serum was evaluated at the Gluck Center, the Gluck Equine Research Center by Dr. Chambers' lab, and the investigators were all blinded, so they didn't know which group of horses or how these, what vaccines they were administered. So essentially this slide simply just summarizes what I just said. So we would have one group of horses that would get the bivalent, the risk-based influenza and herpes in one syringe. At the same time, they would get the core vaccines in a separate syringe. So Eastern, Western, tetanus, and West Nile. The other group of horses would get basically the core and risk in one syringe. And as I said, we looked at all the different manufacturers and did the appropriate analysis or did comparison of the influenza titers. So the summary of this is that all manufacturers' results trended in the same direction. Okay, so basically the flu-rhino combination consistently, the bivalent, consistently provided a better cross-reactive immune response to their corresponding polyvalent vaccine. So you can see in this particular study, basically we had 37 out of the 40 horses had a cross-reactive titer greater than 1 to 10. In the cross-protective side of the equation, which is a 1 to 20 titer, 34 out of the 40 horses, or 74%, had a greater titer. So basically, what does this mean? So what we found is that really, regardless of the manufacturer, the immune responses were better with the flu-rhino bivalent vaccines than they were when they were given as a polyvalent vaccine containing all the core antigens. The cross-reactivity responses ranges, as I said, the trends were the same for all the different manufacturers, but they range from a minimal different to highly significant. When you get a 0.0001, that's highly statistically significant for you statisticians out there. And the vaccine, again, this was compared to their polyvalent vaccine. So the bottom line is that for really a more consistent immune response when it comes to the circulating influenza strains, our belief in our data is leading us to, or suggesting that the flu-rhino should be administered as a straight bivalent vaccine separate from the core antigens. So with that brief overview, I've got a couple of questions that I'm going to address to Dr. Chambers, since he is the gentleman that has done this, done all the analysis for us. So Dr. Chambers, essentially what is the OIE, the surveillance panel, and what do they really do and what do they recommend?
Mark, the expert surveillance panel is a group of virologists that study equine influenza. We are representing the 4 OIE reference labs for equine influenza, mine, England, Ireland, Germany, and we have also representatives from around the world. Equine flu is a worldwide disease. We have representatives from Argentina, from India, from Japan, from China. From time to time, if there has been an outbreak like in Australia 11 years ago, then we will invite an expert from Australia that was working on that outbreak, because we want to get the as much as possible the first-hand experience and not just what was reported like online. If you read down the slide there, what do? We review for each year. We meet generally around the end of February, beginning of March, and for each of these meetings, we review the epidemiological situation for equine flu for the previous year. We are particularly interested in evidence for vaccine breakdowns, because our charge, you might say, is to make strain recommendations for the influenza vaccines. Just like the human vaccines get updated almost on an annual basis, so we have the similar kind of antigenic drift in the equine influenza viruses, but it doesn't go nearly as fast. The panel reviews the characteristics of the viruses that have been isolated. That is, isolation is a key for us, and so it's not enough to have a PCR positive. If, for example, if Nicola Pusterla is here in the room today, I haven't seen him, but his lab has collected more nasal swabs from horses over the last 9 or 10 years than anybody, and a number of those are PCR positive for equine flu. My lab is in touch with him. He sends us his positives. We try to do virus isolations. We need the isolates for 2 reasons. One, because you can't determine antigenicity from a PCR result. You can't determine antigenicity from a sequence. The sequences are informative, but what we really want to know is antigenically, in other words, how does the horse immune system see this virus compared to the viruses that are in the vaccines? And the sequence information, but also the antigenicity are key, and we need those virus isolates. And so to the practitioners, I would emphasize, please submit nasal swabs, or ideally nasopharyngeal swabs, to your veterinary diagnostic laboratories in event of respiratory disease. The positives, eventually I will get my hands on, if they are flu, eventually I'll get my hands on them, and my lab can try to do the virus isolations. So have I said enough about that?
Thank you, Dr. Chambers. So for the next question is, essentially inquiring minds want to know what part of the virus is important, and how does the OIE, you mentioned this a little bit, conduct surveillance and then make strain recommendations? Those
of you in the room that remember virology, remember equine influenza, all influenza viruses have an envelope with proteins that are sticking out of that envelope. These are the hemagglutinin and the neuraminidase. They are the major antigens of the virus. And it's been known for 50 years, more than that, that immune responses directed against primarily the hemagglutinin, secondarily the neuraminidase, are protective. And so there are vaccines out there that consist of nothing but the hemagglutinin and neuraminidase, and the rest of the virus is just sort of along for the ride, or even fractionated out. There are other vaccines that are the intact virus, but it's still basically the hemagglutinin and secondarily the neuraminidase that we study. Those are the determinants of the antigenicity that differentiate one strain from another. Now, the standard technique for measuring antigenicity is a test called the Hemagglutination Inhibition test, or HI test, and it's also been used for over 50 years. But it really shows its age. It has limitations. One of the big limitations is comparing HI results from different laboratories that were using different test antigens, and trying to make sense of all this HI data. And so what is shown on the screen is a fairly new technique. It's a computerized interpretive device for making sense of large masses of HI data. It's called Antigenic Cartography. But what it amounts to is the viruses that are antigenically similar will map to some sort of cluster. And you can see there's 3 clusters on the screen. They correspond to the Florida clade 1, the Florida clade 2, and the old Eurasian lineage. The key is as long as viruses still map within a cluster, then they are similar enough that one representative strain from that cluster ought to be protective in a vaccine against everything else in that cluster. Okay, so Ohio 03 has for years now, much longer than I thought it would, be the representative strain for the Florida clade 1s. And Richmond 07 for the last 10 years has been the representative strain for the Florida clade 2s. The Eurasian strain lineage, we've not had an example for about 10 years now. But what we are on the lookout for are development of new clusters. I mean, this is not, these particular clusters are not going to last forever. Eventually there's going to be an outlier that starts to become a dominant circulating strain, and then viruses will map to that outlier and form a new cluster. And that's what we want to be able to detect as it's happening. The development of a new cluster is the signal to us that we need a new vaccine strain that will protect against that new cluster. And we haven't seen it yet. So that's why Ohio 03 is still in the vaccines.
So, all right, well, good. Well, thank you, Dr. Chambers. We're going to come back to this. So if you've got questions, hold them. I want to get through the other study that Dr. McKenzie is going to present. This is a study that was just recently completed at down in Virginia. And take it away.
So just want to share with you all a study we performed last winter, as Dr. Crisman said, in Southwest Virginia. For obvious reasons, we performed the study in the winter to try and avoid natural exposure during the course of the study that might have interfered. And we were really just asking the question of, is there any difference in immunogenicity between the commonly used Potomac Horse Fever vaccines, which is basically, there's the monovalent Potomac Horse Fever vaccine that you give on its own, either with or without other vaccinations. And then there's the multivalent combination Potomac Horse Fever rabies vaccine. And so we had 91 client or university-owned horses that participated in the study. 45 of the horses received the monovalent vaccines, so they received Potomac Horse Fever as a single vaccination and rabies as a single vaccination. They were given 4 inches apart on the left side of the neck. And then we had the 46 horses that received the multivalent at a single site on the left side of the neck. And prior to starting the study, we screened all the horses a month before so that we could create our groupings for the analysis, so taking into account age, sex, barn, those kinds of factors. And then when we began the study, we pulled baseline titers immediately prior to vaccination, and then we titered everybody at 1, 2, and 3 months post-vaccination. The IFA titers were all performed at the Cornell Animal Health Diagnostic Lab. We ran the screening titers as a single batch, and then for the study itself, we saved all the samples frozen and submitted them all as a single batch so they could be assayed using the exact same reagents, so we wouldn't have inter-assay variations. It's probably not a shock, if any of you who've dealt with Potomac Horse Fever, to learn that both vaccination protocols exhibited poor immunogenicity. We've known this for a long time. It does not necessarily mean, obviously we get some benefit. It doesn't mean that they don't provide benefit, but we just don't get a strong immunogenic reaction to the vaccine. And indeed, in this study, only 1/3 of the animals demonstrated seroconversion at any time point after vaccination. Now, there's not published data on what it means to seroconvert, but what we used as our marker for our qualifier is a greater than 400 increase in IFA titer was considered to be representative of seroconversion. We did see that the monovalent Potomac Horse Fever vaccine exhibited significantly greater immunogenicity both in terms of the number of horses that seroconverted after vaccination and in terms of the magnitude of the immunogenic response that we saw. And that was significantly different at 1 month and 2 months post-vaccination. And so you can see here, at 1 month post-vaccination, we had almost 25% in the multivalent group who seroconverted, and about 44% seroconverted in the monovalent group. And at 2 months, it's 20% and 40%. And then at 3 months, it was not significantly different, at still around 20% and 30%. Obviously, the response in the monovalent group was sort of deviating back towards the mean, so those titers were decreasing over time. And then if you look at the magnitude of the serologic response, and this is mean IFA titers, at 1 month post-vaccination, you can see there was a significant difference in the magnitude of the titer. In the multivalent group, titers went up from around 250 to about 500. And in the monovalent group, they went from basically the same initial value at baseline to up around 1,200 on average at 1 month post-vaccination, and then decreased to around 900 at 2 months post-vaccination. And similar to the seroconversion data, the serologic response for both groups was essentially the same by 3 months post-vaccination. And so the monovalent vaccine also exhibited, if we looked at median IFA titers, which was what was used in the statistical analysis for the publication, which is under review, that was also significantly different. And as I've already said, there was no significant difference between groups at 3 months in either seroconversion rate or the median IFA titers. I'll turn it back over to our MC, Dr. Crisman.
All right. Thank you, Dr. McKenzie. Dr. Divers, the first question I have for you is, how should you approach a fever of unknown origin in a practice area that is endemic for Potomac Horse Fever near Rickettsia?
Well, you're probably going to treat the horse with oxytetracycline, number one. If you're in an endemic area, it has anaplasma Potomac Horse Fever, but hopefully samples would be taken to try to confirm what the diagnosis is. And of course, those appropriate samples for Potomac Horse Fever would be blood and fecal PCR combined. Now, we sometimes will recommend saving serum just in case that both fecal and blood PCR are negative and everything else is negative, such as anaplasma, et cetera. Because sometimes then you can go back and collect a second serum sample and show seroconversion when actually the PCRs have been negative. Now, that's not particularly common, but it definitely does occur where we'll have negative PCRs, fecal and blood, and then the horses will have seroconversion, so we confirm it that way. But that's kind of the backdoor way to diagnose it. And then an anaplasmosis would be the other known common pathogen, which is frequently in similar areas that you find Potomac Horse Fever. And of course, that can be response to treatment with oxytetracycline. And then PCR on the blood for anaplasma would be the way to diagnose that. And sometimes if you do a CBC and you see a thrombocytopenia, a horse with fever of unknown origin, in endemic areas for Potomac Horse Fever or anaplasma, if the CBC demonstrates a thrombocytopenia, that's almost diagnostic for anaplasmosis. And then, of course, there are other agents that we probably don't know about, and including we know about the viral respiratory agents, but there are probably other tick-borne agents that cause fever of unknown origin, and those are more difficult to track down. I mean, you can spend a lot of time and a lot of money trying to track down some of these fevers of unknown origin, but rule out the common ones first. Potomac Horse Fever, anaplasma, viral infectious agents. And then if you're having a problem in your practice area, you might want to talk to somebody at the diagnostic lab to try to track down by molecular techniques, maybe some of the less frequently recognized infectious agents.
The next question is the Potomac Horse Fever geographic zone or area expanding? And basically, is the incidence of the disease or the season of year for the disease increasing? And you don't have to talk about climate change.
All right, I promise it will not. Yes. Okay, can you hear me now? Okay. So as far as the geographic range for Potomac Horse Fever increasing, we don't have good data on that, but based on individual case publications, which you'll occasionally see in the literature, it might suggest that it is increasing the geographic range. For example, occasionally you'll see an article from Manitoba or Saskatchewan or somewhere where they have recognized the first case of Potomac Horse Fever. And so it's possible that it is expanded, but we don't know that for sure. It may be just the recognition is occurring. And then the second part of that question was?
Was the season of the year.
Oh, the season of the year. Well, it's hard to talk about that maybe without talking about climate change, but the season of the year, certainly in the Northeast, I mean, we see cases in October. In Kentucky, they have confirmed cases in November. And so the season is quite expanded. We'll see Potomac Horse Fever really anytime from April into November. And I don't know whether that's expanded once again, but I think it should be on the radar as a cause of fever of unknown origin or diarrhea and toxemia anytime between April and into November.
So keep that thought, and you've kind of alluded to this on the first question, but really what is the best way to diagnose Rickettsia or Potomac Horse Fever?
Well, once again, it's a combination of submitting samples. Well, number one, know that it's in your endemic area. It's endemic in your area or occurs in your area. So knowledge of that's a good way to start. And then ruling out other diseases, but if you want to go straight to Potomac Horse Fever, it's a PCR in blood and feces combined. And I think some people don't realize that, but it may be positive in the blood, negative in the feces. It may be positive in the feces and negative in the blood. So if you really want to track down a diagnosis, you should submit both those samples. And once again, occasionally you might want to even save serum from the horse in case you need to go back and check another serum sample 2 weeks later if everything comes back negative to try to help confirm this Potomac Horse Fever.
Okay, and then I guess, for you now, how important is the disease in endemic areas? And what is the estimated expense and mortality in infected horses?
It's estimated that only about 20% of the horses that get infected have clinical disease. So you think then it may not be that important disease, but it is. And the reason for that is both mortality and expense in treating these horses. Because if they are not treated early enough with oxytetracycline, which works quite good against the organism, if they develop clinical signs of colitis, then those horses often require intensive therapy, often in a range of sometimes thousands of dollars in order to treat those horses with fluid therapy and other treatments for that. And the other big problem we have, of course, is laminitis. Seems to have a very high risk. The horses with colitis, and sometimes horses don't even show diarrhea. They'll have just fever. Occasionally proteins will drop, as in protein-losing neuropathy. They'll have toxemia. They may not ever develop diarrhea. But many of these horses will develop laminitis. And of course, that's a costly disease, and that's a disease that once it occurs, we don't have a magic bullet to give those horses to actually allow them to go back to normal structure of their hoof and even normal function. So it can be, when it does occur as a clinical disease, it can be very, very expensive to treat, and you have a mortality in some cases due to toxemia. And then the biggest problem we have is those horses are founder. We have so many horses, we spend a lot of money on treating the diarrhea disease, maintaining hydration, treating toxemia, and they get through that. But if they founder along the way, we find out sometimes they get euthanized once they return back to home property because of laminitis. So it's a very frustrating disease to work with.
Okay, so Dr. McKenzie, I don't want you to nap over there, but based on the results of the study that was done, basically what would be your recommendations for vaccination? And Dr. Divers can help you on this too.
Probably need some help. I think the seasonality is really the biggest challenge, and so just to kind of add to what Dr. Divers said, for this study we actually started out with 100 horses, and we eliminated 9 because they seroconverted in November. And so none of them were clinically ill, but some of them had titers go up as high as 3,200 from very low baseline titers. So we were having exposure in the mountains of Southwest Virginia in November. So that is not supposed to happen. It's much more of a long-term problem. You know, when I was up in Maryland, we were very focused. You know, we're focused on the summer, but I think we need to make sure we've boosted them early enough in the spring that we can catch sort of those early April, May cases, and then we probably need to be boostering towards the end of the summer. You know, as the data showed, we really only have a serologic response for about 3 months. And so, you know, probably want to hit them again, say August or so, to get that real good booster response, because that's really peak disease time, is that late summer and now well into fall and almost into winter. Depending on where you are, it could be a year-round problem. And so I think still 2 boosters is probably appropriate, but maybe spacing them out a little bit more, trying to catch them more the second booster later in the year.
Okay, so when it comes to, and this is for both of you gentlemen, to protecting against Potomac Horse Fever, is there anything else specifically that you can recommend? Drain the swamp?
Well, I mean, there are management tools. We've had a couple of big operations over the last few years that have had sort of mini outbreaks. And, you know, we always used to regard Potomac Horse Fever as sort of, you know, the one horse on the farm might show disease, but we're talking 5, 10 horses on a single property. And when we've gone and looked at it from an epidemiologic standpoint, there are some major risk factors like lights. And so you'll walk onto these farms and there's just a carpet of dead aquatic insects underneath these lights. And so keeping lights on, particularly in areas where the horses are really spending a lot of time, probably increases exposure to those aquatic flies when you're having that heavy hatch that's bringing the disease from the water to the facility. And so that's one thing that we've recommended is, you know, maybe turning off some of that night lighting, at least in the areas where the horses, you know, we had one where there was a light right over the water trough. And so it was just, you almost couldn't see the water for all the dead flies floating on top of it. So simple management changes like that, I think, can be helpful in addition to vaccination. I don't know if you have any other thoughts.
Yeah, the only other thing I've noticed over the years that, you know, you'd think with drought conditions we may not have this problem as much, but we've seen outbreaks during very dry seasons of the summer. And I don't know with certainty what's happening, but I have noticed in some of these farms I've been to that although the grass is pretty scant due to the drought conditions, there may be a wet area, one wet area in the pasture, and it makes me think that maybe these aquatic insects are probably congregating in these wet spots in the pasture, and therefore all the horses are probably being exposed to it because those wet areas are also going to be the areas where you still have grass to eat. But when I was at Pennsylvania, I noticed a couple farm outbreaks I went to that I observed that. So I think the bottom line is if I were in an endemic area with a lot of Potomac Horse Fever and I had just these wet areas in a pasture, I would try to somehow have the horses avoid those if possible.
Okay, and some of you may not be aware, but we just had I guess it was a couple months ago in Lynchburg, Virginia, we had a big outbreak of PHF in 7 horses at their equestrian center. There is not a natural water source, but they did, as Dr. McKenzie said, they had a common water trough outside in their paddocks and they kept these big floodlights on at night. So the suspicion is that it may have had to do with the lights, but it was 7 horses. And I believe 2 of them died. So anyway, so we'll hold that thought. I'm going to have a few sort of conclusion statements here, and then we'll open it up to the audience, to the group for extra questions. So basically, the overall summary of this is that interference with these multivalent or polyvalent vaccines has been observed, but we do not know what the mechanism is specifically for that. This has been looked at, as I mentioned, specifically in pediatric vaccinology, so the FDA has been evaluating this for decades. Some of these combinations, for example, the MMR, the measles, mumps, rubella vaccine, is a polyvalent, but it works the same. It satisfies that definition up there that it does generate the multivalent vaccine does generate an immune response to their individual constituents as monovalent's the same as a polyvalent. But interestingly, if you add varicella to that combination, to the MMR, that combination, it goes out the window. So something happens, and they don't know what it is. There's references to polio and rotavirus vaccination that is an oral vaccine that has demonstrated interference, cholera and yellow fever, so there's a whole host of these. On the cattle side of the equation, they've looked at Pasteurella hemolytica and bovine herpes type 1. So there you're mixing a Bactrin with a modified live. Again, there's a problem there. And then we've demonstrated the studies with the influenza and the PHF and rabies, where the PHF administered as a monovalent does provide superior or better protection than it does when it's administered in the bivalent combination. So basically, decreased immunogenicity in these vaccines. When multiple antigens are administered in a single syringe has been documented both in human and veterinary medicine. As I said, we do not know what the mechanism, and it's probably multifactorial. And it's probably pretty complex. So it could be due to maybe pre-existing immunity, could be a reflection of the adjuvants that are contained in the vaccine, could be pH. Said there's a whole host of factors that are involved in this, and certainly interactions between viral components and or Bactrin components have been reported, as I mentioned with the bovine herpes type 1 and the Pasteurella vaccine. So basically, this overall interference generally decreases immune response to one or more of the antigens that has been administered. So what we've done, and I do want to make this clear, and you all are probably familiar that Zoetis has just come out with this core EQ innovator. This is not a big combo. Oops, what did I do here? This is not a big combo. This is basically, it contains the core antigens. So it's Eastern, Western, tetanus, West Nile, and rabies. So essentially that's what we've done. So again, when we protect against all these core diseases, you add the risk-based vaccines as you see necessary in your particular site. So obviously, if strangles is a problem, or flu rhino is an issue, or Potomac Horse Fever, these vaccines, these risk-based vaccines, should be added into your protocol individually as you see fit, and also associated with the seasonality of those specific diseases. So separating the core and risk antigens does provide some flexibility and certainly efficiency in our vaccine protocols. So with that, we've got a few minutes. If anybody has some specific questions, we're happy to lob the cube in your direction.
How far apart from giving the core vaccines? Nobody knows the answer to that. I think you should give it as a separate shot. How often? Flu is a risk-based disease. It's not a core disease. And so for just a pleasure horse that grazes around somebody's backyard, probably once a year might be enough. For a 2-year-old in training that goes to a training center, goes back home to the farm, then maybe out to a race, then the young horses in particular have for their primary course of vaccination what we call the immunity gap. And we played around with trying to shrink the immunity gap. The reason there is a gap is because in the young horses in particular, you give them the vaccine, they'll get a antibody spike, and then the antibodies start decaying almost immediately. And 2 months later, you're back where you started, okay? So the higher you can make that spike, the longer it'll last before the young horse becomes vulnerable again. So a young horse, think about what your plans are for that horse and when you could expect that it might be exposed. And so you might vaccinate a little more often than every 6 months. Older horses, their antibody titers tend to be more stable. And so every 6 months might be adequate for a horse that's 5 years old or older than that.
So, and I think I got a clarification from Dr. Voris. He's my translator. If we're talking locale, when we did the PHF rabies study, we did them on the same side of the neck, and we separated them by 4 inches. So some people you can give them on, but you can give them at the same time, and you can separate them by a few inches. Okay, is that kind of what you were asking? Oh, you asking, so you got everything, okay. Same price. Same price, yeah.
Older horses generally don't travel very much, and so you would probably assess their risk as being low. You could, if that's the case, and if they're not like sharing a paddock with horses that do travel, but they're in, with, their age group, basically, that would be a low risk group, and so they're probably not going to have much exposure. So maybe once a year might be adequate for a low risk group.
So, yeah, I mean, I think it's a very valid point. And, the problem is we don't have good markers for cellular immunity. The ideal thing would have been to do actually a clinical trial, but given the sporadic nature of the disease and the frequency of subclinical infections, it really wasn't within the scope. And so in this study, we're just using serology as a surrogate marker of immunity, and absolutely it does not tell us definitively about protection.
And I kind of look at PHF, and I know a lot of people may not use the vaccine even in areas that have PHF, because it's been certainly thought by most veterinarians that it's not probably our best protective vaccine that's available. And you might see one of the reasons why from the data that Harold just presented. But on the other hand, we do know that some horses respond, some horses make an antibody. We do know it's an intracellular organism, so it's no doubt that the antibody's not, the catch-all for everything in protection. It's probably more cellular immunity that would be protective, and it'd be nice if we had a vaccine that produced cellular immunity. But I look at it like rotacoccus, because if you have antibody, you might prevent the organism from becoming intracellular. And that same thing with the equine rotacoccus plasma, and that's probably how it works. It doesn't work once the organism becomes intracellular, but it may enhance neutrophil phagocytosis of the rotacoccus before it becomes intracellular. And so I look at PHF the same way. And because it can be such a devastating disease, I still recommend vaccination in endemic areas, because you hope number one, the horse is going to be a responder to the vaccine, and that's a big concern, because you don't know which horses are responders and non-responders with humoral immunity. And number two, the horses that do respond and get elevated IgG against the Rickettsia, the hope is that will prevent that organism from becoming intracellular and then producing this devastating toxemic disease in some cases. So I'm still a proponent of the vaccine in endemic areas. Yeah, I don't think there's good statistical data on it, so we'd only be guessing, but I would agree. We have some years we have a lot of PHF cases we know about and have in the hospital. We have other years where either they don't show up at our hospital because of cost or some reason, or it's just not occurring as frequently in those years. But yeah, we do see that same trend some seasons. And it'd be really nice to look at climatic conditions if we could prove that's true, to see if, like it's been done with lepto, that maybe climatic conditions could have an effect on incidence in a particular year.
And Dr. McKenzie, I was going to say, Dr. McKenzie was at ground zero when PHF was invented. Yeah, so he probably has some insights in this too. So
The other thing we haven't really talked about is strain variation. And so there are multiple strains of Potomac Horse Fever, so at least 15 that have been described in the literature. And I was talking to a colleague yesterday who's actively doing a study, and they've identified as many as 20 strains in that study, and that data is not published yet. But so I think that's part of it too, is we have the climactic change, and then if you've got horses who, what we used to see in Montgomery County was there were horses on those farms who'd never been vaccinated, and they would spike fevers and get depressed, and if we titered them, they'd have sky-high responses. But they just saw this every year, and they dealt with it, and they had long-standing immunity. But then it was 94, new strain appeared to be active, and we had a horrendous summer. We had multiple cases, and actually Dr. Dutta published a paper from samples that were pulled that year and identified multiple strains that were circulating at one time just in that small geographic area. And so I think that plays a role as well, because we don't know how protection carries across these strains.
And the vaccine was actually a construct of the original isolates from, I guess, what it was the late 80s, early 90s. So that's, again, the organism has evolved, unfortunately.
Can I ask a question on the study? Do we know in the PHF rabies study, do we know if the antigenic mass against the Rickettsia is the same in the two vaccines?
We don't. It's an excellent question, though.